Hello, this is CJ, and in this video, we are going to tackle the different milestones marking shifts from biology to molecular biology. So I hope you get that in this video, we're going to talk about the different events that brought this broad study of life to a more detailed, to a deeper way of understanding life. Okay, so basically, we're going to enumerate different innovations that were discovered, that were made through time regarding the study of life. It's because and the understanding of life went from just basic knowledge of what characterizes living and non-living things to a deeper understanding and utilization of various systems that make up life. So from just looking around us and saying, that is a flying being, this is a terrestrial being, oh, that one is a swimming being. From mere generalizations, mere and basic generalizations about life, that anything that can move is alive, that anything that can reproduce is alive. We are now looking at life molecularly. Okay, we're not no longer just settling for the bigger picture. We're actually going deeper. And now the following are going to be are actually the milestones, okay, of the study of life. They are the markers of our journey from merely observing to utilizing and maximizing our different biological processes. So, yeah, let's begin. All right. For the first one, we have inheritance and evolution. Our conception of inheritance and evolution started at around the 1800s. So pretty much by now, actually, since it's already 2020, stories of Charles Darwin's finches, such as this, ayan. So yung mga finches na na-observe niya by going to an island called Galapagos, if I'm not mistaken. If you are teaching in grade 10, just like me last school year, you would know exactly what I'm talking about because it is one of the key features of evolution and inheritance sa grade 10 biology. So it is basically a short discussion about how birds, despite the fact that they are of the same species, almost the same structure, they tend to have some differences, such as in the shape of their beaks, kasi iba-iba yung diets nila. Ayan, as shown. And also the story of Gregor Mendel is, you know about it. He tended to these garden pea plants and he observed several things. He compared them. He also noticed some differences. Pretty much all of his work paved the way for the traditional, the earlier, the first or the oldest, okay, version of genetics, which is yun nga, Mendelian genetics, and he's the daddy of genetics that made him the daddy of genetics. Yes, basic stuff, tapos naging mas masalimuot, mas naging detalyado lang yung studies natin. Now, the other one is Alfred Wallace's wide traveling naturalist studies. He was this person who was really going places, and their stories, Charles Darwin, Gregor Mendel, and Alfred Wallace's voyages. All of these stories they became actually common lore, so common stories in and outside the world of biological sciences. Even though you are not a biologist, you are familiar about these stories. But because they are basic, they are easy to understand, they are easy facts to remember, but their long reaching conclusions, okay, helped us spur the explosion of growth in biology for the last 170 years because of these basic stories, basic discoveries na actually included ang mga nasabi ko kanina. We were able to carry out further studies, and the study of life had this spurt for 170 years, almost 2 centuries. And while it would take the discovery of the DNA, So, ayan si Francis Crick at si James Watson. Pero I would like it to focus more on the structure of the DNA. In the 1950s, to sow the seed of genetic evolutionary studies, we all owe a debt to these naturalist founders who laid the groundwork for many of these things we now take for granted, actually, while conducting our research. So, um, the basic studies now, they have caused the being of some more complicated studies. And so that is the first one, inheritance and evolution in the 1800s. The next milestone is on antibiotics. This is around 1928, so medyo matagal na to. I mean, medyo mahaba yung difference in time. So, you guessed it right. When I say antibiotics, who's the first person to come into your mind? It's going to be Alexander Fleming. So, Alexander Fleming was not setting out in 1928 to revolutionize biological sciences. As you know, his discovery is a product of serendipity, or accidental good things ba laging serendipity. Pero yeah, that was a word we used when I was in high school, the first time that I heard of the name Alexander Fleming. He discovered that something in the penicillium mold spores was able to kill staphylococcal bacteria in a petri dish. As is often the case in science, discoveries make remarkable impacts on research that are totally unrelated to the field they were created to help. Fleming was just trying to find a way to prevent anaerobic infections from being so deadly, not looking to find the world's first antibiotic. If you're gonna ask me, that's actually that's actually better. Yeah, I mean, it's the first antibiotic in the entire world. But along the way, the discovery of antibiotics has been utilized in innumerable research as selection tools in transformation and cell culture, as well as a host of other fields and studies. So, because of his discovery of an antibiotic during that year, we have now a new milestone in the biological sciences. So, thanks to Alexander Fleming. The next one is gel electrophoresis in 1931. So that's the third one. It is difficult to imagine labs actually without the ever-present bench of gel rigs, either humming with the sound of electrical current, dutifully separating proteins, DNA or RNA, or sitting vacant and patiently waiting for another agarose or acrylamide gel. It is just as remarkable to realize that electrophoresis, as we know it, was discovered in 1931 by Arne Tiselius. And even earlier work was done in the early 1800s that provided the groundwork for the Tiselius apparatus to differentiate between proteins. But it was not until the 1940s that scientists started using gel matrices to separate compounds into discrete bands. And it was not until the 1960s that gel electrophoresis would really be used to start identifying DNA and other biological molecules that would give birth to the field of molecular biology. The fourth one, it may not be she. Okay, I was about to use the pronoun she. It's because we're going to talk about something that belongs to to a female. So it may not actually be a common knowledge. So medyo kakaiba to. Okay, because the person who actually caused this milestone did not really study about it. It's sort of like ano, it's actually from her, literally. Okay, for you to know what I mean, we're going to talk about Hella cell discovery in 1951. So Hella stands for Henrietta Lacks. So that is Henrietta Lacks. she was a woman. The pancreatic cells taken from her body before she died in 1951 have become a benchmark in the history of cancer research and knowledge. Okay, so her cells were immortal. Okay, HeLa cells are immortal. They made medical research easier, more robust, and repeatable. The cell line is what allowed for the creation of Salk's first polio vaccine in 1952. Since its discovery, there have been over 111,000 patents created involving the HeLa cells. It is safe to say that without Henrietta's cells, a great body of research would have been slower, and biomedical advancements a great deal more ponderous. So she did not really study HeLa cells because they came from her literally. Okay, I hope you get that. Okay, so now moving on to the fifth one, the structure of the DNA in 1952 to 1953. So, as with with the discovery of inheritance and evolution, yung kwento ng discovery of the structure of DNA is also well known. It starts with Rosalind Franklin's first image of the double helix in 1952, and then subsequently sina James Watson and Francis Crick naman ginawa na nila yung model para doon in 1953. Kaya lang, Oswald Avery had already identified actually the DNA way back in 1944, okay, as the primary point for hereditary information, but the structure of the DNA cannot be overlooked because it's very relevant in our understanding of so much of what is now considered common knowledge in biological sciences. Okay, so the point there is, despite the fact that Oswald Avery was able to discover it first, functionally, it is important, equally important for us to, it is, the structure is as important, which I think was not done by Oswald Avery by that time and was done by Rosalind Franklin and Watson Solas Crick. So, yeah. There are also some different some sources. I mean, there are conflicting sources about what really happened between Rosalind Franklin and James Watson and Francis Crick. But as for this video, we are using this kind of this understanding. Okay. Next one. Okay, so the sixth one, DNA polymerase in 1956. So Arthur Kornberg and his lab forever changed the world of molecular biology with the discovery of DNA polymerase from E. coli or Escherichia coli cells. So, yeah, DNA polymerase is pretty much that thing that you know na nagpaparami. Okay, it is the enzyme that is capable of of amplifying, of copying DNA strands. So in one instant, scientists were now finally capable of synthesizing new DNA sequences onto an existing DNA strand. So the use of the original DNA polymerase and subsequent polymerases discovered by Arthur's son, so Thomas Kornberg, and others have created the bedrock of molecular biology regarding PCR, such as cloning and then transformation and sequencing. Without these workhorses of the lab, much of what we currently understand about our DNA and life would be non-existent. Basically, yung pinaka magandang nagawa rin talaga ng DNA polymerase is we have discovered this tool that will effectively multiply DNA samples, because that is pretty much a really big problem before for biologists. Yun. So this video is getting quite long. I noticed that. So next one is reverse transcriptase. So reverse transcriptase was independently discovered by both Temin and David Baltimore. So yeah, so this is Howard Temin and David Baltimore, but they discovered it in the same year, 1970. So as a revolutionary tool, Reverse transcriptase, the enzyme, finally allowed scientists to synthesize complementary DNA and double-stranded DNA from RNA. We all know na in the central dogma, it's the other way around, from DNA to RNA. Pero yeah, it's the reason why it's called reverse transcriptase. It's because it is the enzyme that allows that to happen, the reversal of the central dogma, the normal DNA to RNA flow of the central dogma to happen. So it now bridges the large gaps in knowledge of the character and sequencing of RNA by use in PCR. Now, discovering the roots of RNA and its translation into proteins is a fundamental necessity of biology, and the realization that RNA could be transcribed into DNA was paramount in the understanding of retroviruses and later even antiviral drugs. So yun siya. Um, we are getting I mean more detailed here. So I hope you're still with me because the next ones we're actually getting deeper into the molecular matters of life. So let's continue. The next one, it's for restriction enzymes. So it's in the same year, 1970. The first restriction enzymes were discovered in the early 1950s by Salvador Luria, you know that guy, John Weigel. Yeah, I don't even know if I pronounced his name correctly, and Giuseppe Bertani. But the enzymes they found were all type 1 enzymes that cleaved DNA randomly from a recognition site. In the same year, actually in the 1950s yun, in the same year, in 1970, Hamilton Smith and siya, and associates, actually leading associate is Daniel Nathans, that guy sa may far right, okay, discovered a more popular type 2 restriction enzyme that cleave at their site of recognition. And yeah, they showed that by cleaving in those places, they could separate the fragments via gel electrophoresis in order to map the DNA. Now, the use of restriction enzymes to produce a predictable cleaving pattern to work from has become a benchmark in cloning and mapping. Again, let me repeat that. The use of restriction enzymes to produce a predictable cleaving pattern to work from has become a benchmark in cloning and mapping, because if you are just going to have type 1 enzymes, yung kay Salvador Luria, John Wigley, and Joseph Bertani nga, in 19, the thing that they were able to discover in 1950s, how are you going to effectively clone when restriction is happening ever so randomly? So that was a problem. Ayun. Ayun. So the next one is E. coli transformation. Bacterial transformations have been around since the 1920s. Escherichia coli was being utilized as a model organism in microbiology and other biological fields for most of the 20th century, but was considered intractable to transforming until Morton Mandel and Akiko Higa, na hindi ko mahanapan ng photo, were able to induce it to take up DNA with the use of calcium chloride. Okay, so the discovery of artificially induced competent E. coli cells created one of the easiest and most efficient transforming bacteria, which allows for even simpler cloning methods in all biological science. Okay, so the use of E. coli has only grown into popularity as one of the most model organisms in science, and was one of the first organisms to be completely sequenced in 1977. So take a break, okay? So from the usual acceptance of what I'm actually from the discovery of that there is actually a DNA, a genetic material. Now we are trying to transform some information from, one form, of course, to another. We are capable now of sequencing at least a microbiological organism, that is E. coli. Okay, so now we move on to polymerase chain reaction, or PCR. So a few discoveries have revolutionized their field as much as polymerase chain reaction, or PCR. If you're gonna ask me, my biotechnology professor way back in undergraduate college, and actually even in my masterate, she's always telling me that she always tells us, her students, that PCR is one of the most important discoveries that man has made in the field of molecular technology and even in biotechnology. So likewise, PCR owes its own revolution to the previously discovered thermally stable DNA polymerase. Without that, okay, without the DNA polymerase, or the discovery of the DNA polymerase, actually PCR may not even work. Okay, because will not even come into existence, because what are you, you don't have that concept of multiplying DNA. Okay. So yeah, prior to Karimuli's work in reinventing an enzymatic assay to utilize a DNA template, primers, and heat cycles, first described in the Journal of Molecular Biology in 1971 by Gel Clip, cloning was slow and very tedious. And even in the early days of PCR, the heat cycles would denature the polymerase, requiring it to be added anew every cycle. So PCR may be the single most indispensable technique used in modern biology. So yeah, we get that answer. It is. If it's not the most important, it's one of the most important discoveries in modern biology. Okay, next one you have bioluminescent markers. Bioluminescence has been observed for millennia, but the understanding of its nature and the reaction that produces it remained a mystery. In 1955, Osamu Shimomura was the first person to crystallize luciferin from ostracods, and was later instrumental in the discovery of GFP in jellyfish. Later on, we'll talk about GFP. Firefly luciferase was finally cloned in 1985, but the use of bioluminescence as a marker really began in 1986, when it was first utilized as a gene marker in both tobacco and Arabidopsis plants. So that's a tobacco plant, that's a growing tobacco plant, and it's glowing. Yung marker gen are bioluminescent. And by 1988, it was being used in mammalian cell lysates as a prominent tool for in vivo studies of gene regulation. Bioluminescent imaging is still one of the most widely used applications in both in vivo and in vitro research in nearly every biological system. Okay, so the next one we have gene therapy. This was in the 1990s already. So gene therapy had been science fiction for most of the 20th century. Something that is only found sa mga stories, a nearly magical way to cure genetic diseases. However, in 1972, Theodore Friedman and Richard Roblin, first introduced the possibility that it might become reality someday. Even if they believed that humanity needed to be extremely cautious about taking that giant leap into the unknown of genetic manipulation. But by 1990, William French Anderson was given permission by the US National Institute of Health to conduct a clinical trial for a patient with a severe immune system deficiency. Cancer gene therapy trials were approved by 1992, and many other genetic disease therapies have been conducted in the decades since. While gene therapy remains a miraculous opportunity for many of our worst genetic diseases, there is also much to be concerned with over its possible misuse and the ethics surrounding it. The next one, if we had bioluminescent markers, we also have fluorescent protein markers. Okay, so ngayon natin pag-usapan yung GFP. Along with bioluminescence, fluorescent markers have made an equivocal impact on research. Perhaps it is appropriate then that Osamu Shimomura would have been instrumental in discovering both actually. So GFP stands for green fluorescent protein, and it was first discovered by Shimomura in jellyfish in the 1960s along with the blue acurin protein. Later, though, Douglas Prasher utilized GFP and reported its genetic sequence in 1992, which allowed it to be expressed in E. coli in 1994 and later in C. elegans. The use of bioluminescent and fluorescent markers has let us visualize the mysteries of the cell, protein-protein interactions inside, outside, and even in between cells. We can see how cancers react with cell lines or even inside the bodies of experimental animals. Now, due to the enormous range of colors that species have developed through evolution, we now have the capability to understand the magnitude of microscopic interactions that were only imagined before. The next one is RNA interferences or RNAi. Scientists had been aware of a system of co-suppression or quelling for quite a while. Plant biologists had been aware that sometimes overexpression of genes in plants to create more vibrant colors would unexpectedly produce plants with variegated color patterns or even no pigment at all. In 1998, though, Craig Mello and Andrew Fire, okay, that's Andrew Fire, published their work documenting the intentional silencing of genes in C. elegans via a new process called RNA interference, in which they combined both a sense and antisense sequence of a gene. The process would evolutionarily be used to defend against viruses that try to insert themselves into the DNA. The use of RNAi has become critical in the development of gene expression and suppression, in identifying components of cellular processes, and as practical tool in many other biological fields. Don't worry. Before we move on to the last one, let us have a review. Okay, so for the first entry, inheritance and evolution, the main point of this is that we had this very early understanding of the world, that is observing patterns of inheritance through phenotypes, observing evolution by looking at different examples of animals that are expressing some kinds of traits. So that is the first milestone. We had that understanding. Then the second one is antibiotics. We discovered that there are actually organisms capable of producing something, a substance, that can do something really remarkable, such as, say, kill bacteria, in the case of antibiotics. The third one, we were able to isolate different parts of a particular biomolecule, such as a nucleic acid, into its component parts through gel electrophoresis. Through the next one, HeLa cell discovery naman, we were able to conduct so many researches by using cells, actual human cells. So without actually Henrietta Lacks' cells, we will be finding it so hard to conduct trials of medicines, to conduct research on anything actually biological. That's why with our cells, researches became more efficient. The next one is the structure of DNA. It is important for us to know what is the structure of DNA, because without knowing that it is actually double helical, that it is comprised of matching nitrogenous bases, without knowing about the phosphate backbone, and without knowing about the pentose sugar and the other parts of the nucleic acid, we are not going to be able to use it. As what I always tell my students, before you would be able to use something, you have to understand it first. So by understanding, we begin with the structure. The next one is DNA polymerase. conducting researches about a particular thing is difficult. It's more difficult. It's going to be even more difficult if you only have a limited amount of sample. Okay, so through DNA polymerase, we now discovered that it is possible for us to have copies of a single of a limited amount of DNA. Right? It's through an enzyme called DNA polymerase. And through the reverse transcriptase in 1970, we are actually able to trace the source DNA of a particular RNA. Central dogma is always working like from the command DNA to the product RNA. But with the discovery of reverse transcriptase, we were able to trace the product from the command for the product. Okay, so the next one, restriction enzymes. We were able to cut DNA into specific parts or into a specific area. It is a anyway, so yung mga site ng DNA na kailangan lang natin talagang pag-aralan, kailangan natin silang i-identify, kailangan nakakat sila. Okay, so without cutting it into the specific parts, it will be very tedious for us to study DNA. Okay, so the genetic material. And so the next one is cell transformation. From merely accepting the genetic material as it is, we were able now to transform it to transform even the entire microbiological living thing, into something else through E. coli cell transformation. So it is also it's also remarkable that E. coli is the very first organism, or one of the first organisms, to be completely sequenced by the year 1997. Now, following the discovery of DNA polymerase, is the polymerase chain reaction. So the discovery the understanding of or the discovery of the relationship between various temperatures and the actions of DNA polymerase. Ayun sila, yung ganon. Okay, the discovery of that led to even improving our biotechniques. Okay, so now we are not just going to use DNA polymerase and produce limited amounts of copies. We can actually control the action of DNA polymerase by subjecting it to different temperatures, okay, para mag-denature, mag-anneal, tsaka mag-extend through PCR or thermal cycle. Next one, bioluminescent markers. We are using them to as expressions of various characteristics. So bioluminescent markers and fluorescent markers they are aiding in the field of biotechnology, because without these two, we are not going to have an easy way of identifying whether or not a particular trait is present. Next one is gene therapy. So we were able now to understand what genes are. We are using it now for our own benefit. So especially in the fields of immunodeficiency. So it's through gene therapy we can start making the body defend itself. So parang ganun siya. RNAi now is RNAi enabled us to restrict a particular part of the RNA. So we can control expressions, expressions of traits by restricting some parts of the RNA. Okay, I know that is a really, really long explanation. It's already 34 something minutes. The last one is CRISPR-Cas9. This was discovered in 2012. So, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, whereas Cas9 is associated protein 9. So CRISPR was first described unknowingly in 1987 by Yoshizumi Ishino. However, it was not fully characterized as CRISPR until 2001 by Francisco Mojica and Ruud Janssen. The magnificence of CRISPR as a gene editing tool finally came about when Jennifer Doudna and Emmanuel Charpentier re-engineered the Cas9 endonuclease and showed that the new system could be programmed to target any DNA sequence for cleavage. But the future and real value of CRISPR-Cas9 might lie far beyond its targeted gene therapy in a variety of CRISPR adaptations that we have written about previously. Now, all of these discoveries have affected nearly every facet of biological science and have driven research into new and ever expanding directions. It is always amazing to look back and wonder at the greatness of all these discoveries. Now, at the time of these discoveries, these scientists saw their work as simply solving a problem on their bench to discover the answer to their project. Sometimes, that is both the beauty and the pain of research, that the relevance of any particular result isn't fully realized until years or even decades later. But we can be comforted that our research, even our research failures, can be used to further the overall understanding of the world in which we live. And every discovery we make might be used by someone else down the research pathway to discover more. Isn't that what we are all really trying to do anyway? This is CJ, and this is the Knowledge Catalog. Education Solutions. Education Solutions. It has been a really long conversation with you, and I'm hoping to see you in the next video. Goodbye.